So let me quickly recap some of the main conclusions we made from the equations in the last class. From then we'll go on to apply this to understanding hippocampus and memory operations in hippocampus. So we looked at this extension of Hopfield network, which has an activation dynamics du by dt and a separate weight dynamics. And the activation dynamics has two terms. The inputs coming from other neurons, which has a coefficient b, the inputs coming from the external input, which is as coefficient a, and then the weight dynamics has another coefficient c, right? So since both activation dynamics and weight dynamics is going on simultaneously, although at different time scales, right? To distinguish between storage and retrieval, we need to do something about these coefficients. This is what we concluded in the last class. We have to clearly distinguish between the storage and retrieval stages. And that was pretty straightforward in the basic Hopfield network. Because basically you take the input patterns, apply Hebb's rule, and calculate the weights. So that completes the storage stage. And then you freeze the weights and just use it in the retrieval mode. But when both equations are running all the time, that is not quite simple. So you have to somehow make the distinction by controlling these three parameters, A, B, C. So like we concluded last time, since during storage, the input pattern must be more prominent, so A must be high. The lateral connections must not be prominent because lateral connections are initially random. So the inputs coming from other neurons through the lateral connection, they must not be overemphasized. So B must be small. Then during storage, you have to learn the connections based on the external input. So C must be high. Okay, so that is similarly in the retrieval stage, the external input should not be very important. It should be simply acting like a cue. So the external input weightage must be small, and you already have the information about the patterns and the weights. So during retrieval, the lateral connection should become more important. So B should be high. And during retrieval, the weights are supposed to have already been trained. So the weights are not updated anymore. So C should be either 0 or a very small number. So in summary, we concluded in the last class that during storage stage, you have high A, low B, and high C. During retrieval stage, you have low A, high B, and 0 C or low C. Now we will see how these kinds of conditions do seem to be existing in one crucial part of hippocampus, and according to one of the more prominent theories of hippocampus, and that's how the hippocampus is serving its functions as a memory system. So like I said last time, HM, called Henry Molaison, this was a patient in whom the hippocampus was removed. So he had memory deficits. It is through him that we found a lot of early findings on hippocampus and its memory functions. So HM had two kinds of amnesia or memory loss. One is retrograde amnesia, where he had trouble memorizing events or recalling events that happened immediately before surgery. But older memories were intact. And anterograde amnesia means inability to store new memories. So this is a retrograde amnesia, that is from birth on up to a point before the lesion they can remember, and the duration before the lesion they cannot remember. But they had only retrograde amnesia, they can store new memories, but they cannot just remember older stuff. With anterograde amnesia, they lose the ability to store memories after the lesion. In case of HM, this person had both. So you have to combine both these pictures. So it will look, excuse me, something like this. So this should be added here like this. Excuse me. So one more thing is in this mathematical model, we have seen that weights update gradually, right, adaptively. So the kind of weight update we have seen, it's basically a form of Hebbian learning, and we have also seen that A-B learning has two aspects to it, LTD and LTP, and that has evidence from synapses in the hippocampus. So I think I talked about it in one of the earlier classes. So now let us come to hippocampus. Right? What is the connection between upfield network and hippocampus? Now, hippocampus is basically a system for association because it's a memory system, and we know that human memory is an associative memory, right? And it seems to be able to capture relationships among representations of the same object present in different sensory modalities. So suppose I'm holding a cup, right? It has some visual appearance. Let's say it's a yellow cup. It has certain size, right? It is at certain distance from my eyes, so certain shape, and it has certain feel when I hold it, or when I tap on it, it makes certain sound. So all these different features are combined to create this kind of concept called cup. So hippocampus seems to be able to combine all this and create a representation, a high-level representation of the object, and store that high-level representation and also retrieve it. So how does it do that? Let us first look at a simple picture of anatomy, right? Then we'll go deeper into the hippocampal anatomy. So if you look at mammalian brain or human brain, sorry, it's not a great picture, but so this is the central sulcus, which is a major landmark. It divides the brain into two halves, roughly front and back, anterior and posterior. Now here you have primary somatosensory areas, somatosensory, and SS1. This is secondary somatosensory or SS2. Here you have primary auditory, so-called A1. This primary visual, so-called V1. And if you go here, you have V2 secondary visual, just like, and then A2 is somewhere here, and so on and so forth. So you see that there are three major sensory modalities forming some kind of a triangle. Now, if you go deeper into the triangle towards the centroid of this triangle, roughly, you will go to higher and higher order representations of these three modalities. So if you go deeper in the V1 and the vision from the visual side, you will find more abstract visual representations of the object. If you go deeper from the somatosensory side, you will find more abstract somatosensory representations. Similarly, in the case of auditory. So this loose cluster of areas, cortical areas located here, they are called sensory association areas. Okay, now where is hippocampus? Hippocampus is located inside the temporal lobe. It is deeply buried here. So it's not on the surface. So please remember that it's deep inside. And entorhinal cortex is somewhere here underneath. Okay, so information from these sensory association areas goes to the entorhinal via two other stopovers. Okay, so the wiring pattern on the anatomy or in the brain anatomy looks very complicated and confusing, but we can draw it in a simplified manner, almost like a deep network like this. Let us say I have a deep network or a neural network which has three sets of inputs. Let's say this is vision, this is auditory, this is touch, and then this input. So you go through a bunch of stages, constructing more and more abstract representations, right? And then after you go beyond a point, you start merging them, right? So and such merger occurs because if you look at somewhere here, superior parietal area, so that is more between the touch and the vision. So here you will find cells which respond to both touch information and visual information. So logical, right? So these are all association areas. Similarly, you can find combinations of these things. So anyway, so you have, let us say, combinations of all of them beyond a point. And these are my sensory association areas. From here, these are all cortical areas. So from here, I now go down, I go towards the subcortical structures. So you have so you see that in this picture, you can see that if you look at the cortex, so forget about prefrontal, and parietal and temporal, you project to two other cortical areas, parahippocampal gyrus and perirhinal cortex. Okay, so that is parahippocampal and perirhinal cortex, two areas, and from there, so that is like this. From there, it goes to one cortical area called the entorhinal cortex, which is located here. And from here onwards, now the fibers go deep into the body of the temporal lobe, into the hippocampal circuit. So from here, it goes to hippocampus. And even hippocampus, again, there is within hippocampus, there's lots of circuitry. And from hippocampus, again, the connections come back to EC. And say in cortex, the general rule is most connections are reciprocal. That is, if an area projects to B, A projects to B, then B returns the connections to A. So these are called reciprocal. So you'll have information going in this direction, and also there's information coming back. So this is simpler to understand. Basically, you have three pipelines, the sensory pipelines. As you go higher and higher, you construct more and more complex abstract representations of the sensory input, and then you also combine them. So if you combine them, you can extract the information of a cup, for example, or a cricket ball, or something like that. And at the higher level representations are now sent to the hippocampus through feed forward and feedback connections. So this is, in summary, what's happening with the hippocampus anatomy. So let me now look at it as in a much more simplified picture. I just call this the cortex. This whole thing I just call cortex without distinguishing whether it is association area or primary sensory or whatever. I just call it cortex. And this blue box, I call it hippocampus without worrying about internal circuitry. So cortex sends information, the sensory information, or this more abstract sensory information to the hippocampus. Hippocampus sends feedback. So what do you think would be happening in this circuit, in this network of cortex and the cortico-hippocampal network? So one conceptual way in which we can look at this is as follows. Suppose I have a cricket ball. I'm holding a cricket ball. It is red in color, round in shape, and hard in feel, and also maybe certain kind of a dull sound if you tap on it. Right. So all these things are now combined, and you send to hippocampus. And hippocampus comes up with a representation, very compact representation, because hippocampus is a small structure. The number of neurons is much smaller than cortex is a huge area. So the small number of neurons, some neurons might respond to this combination of sensory features, redness and roundness and hardness and maybe dullness of sound. They respond to that, right? And so only maybe some neurons respond to that particular combination of sensory input. The thing is, then since you have feedback connections back to sensory areas, whenever you have that set of neurons getting activated, then the feedback connections now in turn will activate the corresponding sensory areas. Okay, what is the utility of that? So suppose I'm holding the cricket ball, I can see the redness, see the roundness, and also feel the hardness. But suppose I'm looking at a cricket ball which is on the table in front of me. I only see the redness and roundness, but not the hardness. But still, let us assume that the appearance of the ball is sufficient for me to activate the same neurons which represent the cricket ball in hippocampus. Then the activation of those neurons and send feedback connections to the cortical areas will further reinforce activation not only in the redness area or in the roundness area, but also in the hardness area. So therefore, what happens is the hard neurons also get activated. This is only a conceptual schematic. Okay, I'm not saying exactly like this happened, but this is roughly what happens. So therefore, what happens is you can now, it's as though you are imagining hardness, although you are not actually experiencing hardness because you're not touching the ball right now. Okay, so like that, the hippocampus acts some people have used the term hash code. Acts like some kind of hash code is like basically a code for an entire record in a database. So the activation pattern of the neurons hippocampus acts like a hash code for an entire complicated sensory activation pattern. Activation pattern in sensory cortex. For that particular pattern, there is a code in hippocampus. You have some other thing. Let us say I'm holding a lemon. It's yellow in color, round, round in shape, soft to feel. That particular combination will activate some other set of neurons. Even if a partial set of the sensory inputs are given, the same neurons will be activated, hopefully. And by feedback, they'll reactivate all the components of that sensory part. This is so you see that it acts like it's acting a bit like Hopfield network. Only thing is, it is more like we can think of it as a bidirectional associative memory. So in Hopfield network, it's a unidirectional associative memory because so in Hopfield network you have right. So this is a Hopfield network. You give the sensory input x at 0. It returns the same vector x after some time, which happens to be the complete pattern. But now let us say you divide the input x into two parts. Okay, let me call this y and let me call this z. So that x is equal to y concatenated with z. Then I can operate this network in such a way that I give y as input, right? Then I recall z. Then I give z as input from the other side. Then I get retrieve y. And this I can do back and forth until both y and z settle down in some stable state. So this is called bidirectional associative memory. It's basically a special case of Hopfield network. In Hopfield network, you present the entire pattern and retrieve the entire pattern. But imagine you've broken up the pattern into two halves, not necessarily have two parts, two partitions. And you give one of the partitions as input to retrieve the other part, or you give the other part as input and you retrieve the first part. Because the Hopfield network doesn't distinguish between address and content, right? Anything can be given as address, anything can be retrieved as content. So this is quite plausible. And this is the kind of thing that seems to be happening. So this is just a schematic that shows how this can be done. Here we have a network here, which has two layers. Basically, a single layer of Hopfield network is now split into two layers. So the full vector v is divided by x and y. X is given as input here, and then it projects to y, and within y then sends feedback to x. Okay, so x0 is given, that gives y0, then y0 is given as input, then that will give you x1, then x1 is given as input, that will give you y1, and so on and so forth. After some time, both will settle down. Okay, so this is called a bidirectional associative memory, and the idea is that hippocampus works as like something like that. Okay, so this is a very simplistic gross picture which just completely lumps then all the cortical areas into one big area called cortex, and the whole complex circuitry within hippocampus is lumped into one box called hippocampus. Now let us expand both of these things and see what they have, what kind of contents they have inside. The cortex, I just talked about the parietal, a little bit about temporal, but another major cortical input, because parietal and temporal are huge. Okay, so that's I don't know, 50% of no, not 50%, but a huge percentage of cortical surface. The other major area which sends inputs to hippocampus is the prefrontal, so somewhere here. So parietal is here, temporal is here. Okay, and all these three areas, so which is pretty high. And you see that all these three areas, what is common among all of them? Okay, so the major thumb rule of brain function is that the posterior brain, back or posterior, is all about input processing. Very, very crude thumb rule, but quite valid. And anterior brain is all about producing output. Right? The output means there are two parts to the output: producing actual output like motor output, for example, moving hands and legs and stuff like that, or moving sublocus areas here, controlling your speech apparatus, right? Primary motor cortex, controlling your limbs and other things. Right. So either actual output generation or just the planning necessary to generate output, all that happens in the anterior brain. In the posterior brain, you have the primary sensory areas like all this stuff which I just talked about, or higher order areas. Okay, so what the areas which send inputs to hippocampus are all at higher level. On the sensory side, the higher order sensory areas project, that is sensory association areas, project hippocampus. On the motor side, it's more the planning areas, not the execution areas, but more the planning areas of prefrontal. This is what these are the parts which send inputs to hippocampus. Okay, so these three areas which correspond to higher level on the output side, which is planning, and higher level on the sensory side, which is higher order representations of sensory inputs. All these send inputs to hippocampus via two other cortical areas, the parahippocampal gyrus and perianal cortex. This in turn projects to entorhinal cortex. And once you got it, like I said, the within hippocampus, so cortex to hippocampus is a feed-forward feedback connection. And within hippocampus also there is a loop, right? From entorhinal cortex, outputs go to a part called dentate gyrus. From there, it goes another part called CA3. From there, it goes to CA1, CA1 to subiculum, and subiculum back to entorhinal. Okay, so this is not the only thing, because in brain, as you take a closer and closer look, you'll find more and more connections. It's very common. For example, there's also a direct connection from entorhinal to CA1. This is called temporoammonis connection, and so on and so forth. Okay, so I'm not worried about mugging up all this connection, but that gives an overall idea. So this picture shows actually a stain version of hippocampal slice, right? So it is there. You have to slice the temporal lobe at certain angle to reveal all this. These are called the hippocampal fields, neural fields, right? So this is because in a real hippocampus, if you open it, cut it open, it won't look anything like this. You can't make out any of this stuff. Okay, so you have to stain it. You will just see neural cell bodies at various places, and you have to work out the connectivity very painstakingly to understand all this network. Okay, so now the same thing I'm showing it in a slightly more biological schematic. The internal cortex has several layers because cortex has 6 layers, right, everywhere, in most places. So the multimodal association areas, the higher order association areas, project to EC, and from the superficial layers of EC, that is 2 and 3. So there are 6 layers. So 1 is the most superficial, 6 is the deepest. So from superficial layers, outputs of EC go into the hippocampal fields, and the deeper layers receive the feedback from the hippocampal fields, and these deeper layers then send feedback back to the higher order cortical areas. So within the hippocampus, and again we have some circuitry which looks something like this. Like I said, from entorhinal, the deeper layers project to dentate gyrus, dentate to CA3. There is also actually a feedback from CA3 to dentate, which is not advertised much, but it does exist. From CA3 to CA1, right? And there's also this direct connection from EC to CA1 called temporoammonis pathway. Okay, so you see that as you see more and more, you'll see more and more connections. Now, you see one branch of EC goes into HC goes to the dentate, which projects to CA1. These fibers are called mossy fibers. Okay, so you have name for every one of these branches. Then another branch EC goes directly to dentate gyrus via the perforant pathway. The mossy fibers are known to be strong, have strong synapses. The perforant pathway has a somewhat weaker influence. So this is a perforant path, and part of the perforant path also goes through the dendrite without synapsing and goes all the way directly to CA3. In fact, that is why it's called a perforant pathway, because perforation means cutting through, right? So this is like a fibers which cut through DG and go beyond to CA3. So then within CA3, what is interesting is CA3 is considered to be a strongly recurrent network, lots of strong lateral connections. Okay, so that is shown by this thick green arrow to show that lots of internal connections. From CA3 to CA1, the wires are called Schaefer collaterals. Okay, so just some names given to all these things. Then, yeah. CA3 is where we need to pay some attention, because this is our Hopfield network sitting deep inside hippocampus. So CA3 has lots of recurrent connections, a lot of internal connections among CA3 neurons. In rat, there are about 3 lakh pyramidal neurons. So these pyramidal neurons are a very commonly found family of neurons in the brain. They are found all over in the cortex and also in subcortical structures. And in CA3 itself, there are about 3 lakh pyramidal neurons. Each of them receiving inputs from about 12,000 other pyramidal neurons. That is, each CA3 pyramidal neuron receives only 4,000 inputs from EC. So the inputs to CA3 neuron come from EC. So a given pyramidal neuron CA3 gets inputs, only 4,000 inputs from EC. But it gets 12,000 inputs from other neurons within the CA3. So external input from EC, these are smaller in number. Lateral connections is much higher. Okay, so also you can even understand this if you take in the ideal Hopfield network. So given neuron receives input from n other or n minus 1 other neurons in the network, but it receives only one input, II, as external input, but receives n minus 1 inputs from lateral connections. But of course, hippocampus is a fully connected network, so therefore you have a lot more lateral connections. But in general, you have a tendency to have more lateral connections than external input, which is natural if you have a strongly recurrent network. So each CA3 neuron receives about inputs from about 4% of all the CA3 pyramidal. This is considered to high recurrence compared to any other part of the brain. And also note that although in the brain there are lots of recurrent connections, it is nothing. It is like nowhere near a fully recurrent network, because if the brain is a fully recurrent network, it has 10 power 11 neurons. If it is fully recurrent, it should have 10 power 22 connections. Okay, whereas it has only 10 power 15 connections. So obviously, so this is correct and this is not correct. Okay, so it's not a fully recurrent network or anything. No. So, but this 4% level of recurrence is very high in CA3. So people have been saying for a long time that CA3 is acting like some kind of Hopfield network, because there is a strong recurrent connectivity, and the other features also which kind of support that kind of perspective. So if you let us take this idea more seriously, let us consider CA3 as a potential Hopfield network, and whatever hippocampus is storing, let's assume that all that is getting stored in this particular network, right? So it must be operating in two modes, storage and retrieval, and by all the arguments that we have applied before, right? If the storage and retrieval is actually happening in CA1, it should work something like this. And again, here I just thought of this simple analogy between the two conditions, the contrasting conditions of storage and retrieval. When I call this the classroom analogy. So in a classroom, let us say during storage, that is, the teacher is giving a lecture and kids are listening and trying to store that. Right? When that happens, the teacher's lecture alone must be prominent. The kids should not chatter among themselves. Right? The chatter among the children should be low. teacher like instruction alone dominates, and the kids are in a state of receptivity, right? So you get to learn the lesson. So teacher's instruction dominate. That means high value of the coefficient A. Chatter among children is low. That is low value of the coefficient B. Eager to learn the lesson. That's high value of C. This is what we want. In the retrieval case, teacher is only asking a question, then kids are discussing among themselves and respond to the question. So in this case, each instruction is low. That means because teacher just asks a question and keeps quiet, right? She doesn't keep on talking. Kids now discuss among themselves, discuss that question and try to respond, right? So therefore, their connection should be high, and so B is high. And there's nothing new to learn, just recalling what they already have, right? So C is low. So this is just an analogy for the two modes that we talked about. So therefore, during storage, external input must be strong, recurrent connection must be weak, and recurrent connections must be plastic. They must be modifiable during that stage. Sorry, this is retrieval. External input should be weak, recurrent connections are strong, recurrent connections are not plastic. This is what we want, right? So now, does it happen in hippocampal CA3 field? The answer is yes, which is very interesting because all the arguments we propose so far are simply based on mathematical considerations. Right? If you want to have a network which acts like an associative memory, and where activation dynamics and weight dynamics is happening simultaneously and all that, then that for those equations to work, you need to have certain conditions. So we didn't really invoke any biology. But you see that very similar conditions seem to be happening in hippocampal CA3 field. So CA3 has two inputs, one EC by perforant pathway directly from EC, other by dentate gyrus by mossy fibers. And perforant pathway fibers are weaker than mossy fibers. That is one thing. Then the plasticity, that is CA3 connection, the synapses are plastic, they are modifiable. And also, so the thing is, this plasticity that we want, right, must be quickly modifiable, right? You cannot, it's not the, so I should, there must be some other higher agent which decides whether the connections in CA3 are plastic or not. This is not the natural plasticity which is driven by external inputs, right? There must be something which permits them. Okay, now you can be plastic, you can change according to your Hebbian rule. Now you cannot be plastic, so I won't permit you to change. So you need some kind of higher agent which controls the plasticity. So now, interestingly, there is a chemical called acetylcholine, which projects to CA3 and other fields in hippocampus. This is a special kind of neurochemical called a neuromodulator. Okay, so neuromodulators, right, mathematically speaking, you need to distinguish between neurotransmitter and neuromodulator. So we looked at neurotransmitters like glutamate and GABA and all that, so which operate at exhibitory and inhibitory synapses. So in this synapse, right, so let us say it releases some kind of neurotransmitter, let's say GABA or glutamate. If you have a neuromodulator projecting to this synapse, so the thing is, synapse has a presynaptic side and postsynaptic side. When the terminal of a neuromodulator neuron comes to this synapse, right, it controls the strength of the synapse by release of a second chemical. So A releases neurotransmitter, then to B, and the C comes in and releases something else which controls the overall strength of this synapse. So in mathematical terms, you can describe like this: A to B, there is a synapse which has some strength, WAB. That strength is modifiable by a third input here, which is neuromodulator. So it's as though you are multiplying this WAB by a factor, which is determined by that neuromodulator chemical. It immediately controls plasticity. Now there are several neuromodulators. Acetylcholine is only one of them, which is plays a prominent role in hippocampal function and therefore in memory functions. And there are 4 major neuromodulators. There are a couple of others like oxytocin and all that, right? And these 4 neuromodulators are different from transmitters in that. The neurotransmitter is like a hotline. It is released by one presynaptic terminal and only heard by the postsynaptic terminal. It doesn't go all over the place. It's a very point-to-point, very precise signal, which crosses the small gap of 20 nanometers. So it's like a telephone call, whereas neuromodulator is like a broadcasting agent, like what you say through a public addressing system. So the neuromodulators are released by small clusters of neurons located deep inside the brain, and these clusters project to these are like loudspeakers. They project to lots of targets within the subcortical structures, and also to a lot of cortical areas. These are really, they punch much higher than their height, sort of. So there are, and usually for every neuromodulator you have two sets of clusters of neurons. One cluster projects to only subcortical targets. The other cluster projects to lots of cortical targets. Like for example, acetylcholine has two such clusters. One is nucleus basalis, which projects to the cortex. See that it's going all over the place. But a small set of neurons sending out long connections to extensive targets all over the brain. The other one is called medial septum, which projects to hippocampus and a few other subcortical structures. Similarly, there is norepinephrine, another important neuromodulator. It is by a cluster called locus cerebralis, projects to lots of subcortical and cortical structures. Similarly, serotonin, raphe-nucleus is a main cluster, projects to cortical and subcortical structures. Then dopamine, released by ventral tegmental area, and I think there's another several. I don't remember all the names. Another one is SNC, substantia nigra pars compacta. VTA projects to lots of cortical targets. SNC projects to the basal ganglia. So these are the four neuromodulators. Acetylcholine is the one which has a lot of prominent role in hippocampal function. Now, what is interesting is blockage of ACH transmission is known to interfere with the memory storage in hippocampus, right? Because if you, so for example, take acetylcholine antagonist, right, called scopolamine. So that will block the action of acetylcholine, right? That is known to induce temporary amnesia. Okay, so for example, so volunteers were given a dose of scopolamine and they were asked to memorize some words, right? And after that, so they were asked to recall the words, and they couldn't recall many words. They just they couldn't store them, couldn't try and recall. And some volunteers even forgot about the trial. They didn't even remember that they went through this kind of a trial. The memory loss was so profound. And I am not sure. This is my knowledge from watching some of the spy movies. People use scopolamines on spies to make them cough up, their secrets and things like that. Anyway, so scopolamine before experiment in this case. So the control subjects who did not take scopolamine, they were able to recall 45 words out of 128 total number of words. Scopolamine people who were right, they could only recall 6 out of 120. So the memory is seriously affected by scopolamine. So, but when the subjects were given scopolamine after the words are presented, that means they're already stored, there's no problem. They were able to recall properly. Right, and there's also evidence that ACH controls LTP in the recurrent connections of CA3. So this first study is at the behavioral level, and that is matched by a more closer study where they looked at the effect of ACH, which controls LTP in the recurrent connection. This will be some kind of electrophysiology study. So Hasselmo and others have developed a theory based on these findings, which basically argues that the CA3 acts like a Hopfield network on the lines of the model that we discussed before. So it turns out that at high levels of ACH in CA3, all these three changes take place. The input is strengthened, recurrent connections of CA3 are weakened, and increased plasticity in the recurrent connections of CA3. Whereas at lower levels of ACH, input is weakened, recurrent connections of CA3 are strengthened, and decreased plasticity. So so if you make an analogy with those equations, A is high, B is low, and C is high. Okay, so A is low here, B is high, and C is low. So exactly what we want. Alright, these two shift between these two modes seems to be controlled by acetylcholine release into ECA3. Now, how exactly does it happen? And again, there's a huge amount of molecular biology, but I'll just talk about a little bit of neuroanatomy or microanatomy that seems to make this possible. So what happens is within CA3 there are multiple layers. So there is one layer here. This is all CA3. There's one layer here, which is called stratum lacunosum moleculae. The second layer is stratum radiatum. The third is stratum pyramidal, where you have the pyramidal cell bodies. Radiatum, where you have radiation. We have lots of wire going around. I don't know what is stratum lacunosum moleculae. It sounds more like a Harry Potter, like a magic spell from Harry Potter. So anyway, so this is where the external input from EC comes in. These are the wires, which carry the intrinsic signals from other pyramidal neurons in CA3. This is where you have the the cell bodies of pyramidals. So when you have high ACH, it suppresses synapses in stratum radiatum, that is this part, more than it does to synapses in stratum lacunosum moleculae, which means it decreases B and increases A, which is what we want. That kind of a differential action seems to be possible because of this nice segregation of synapses correspond to lateral connections and synapses correspond to external inputs. These two sets of synapses are nicely anatomically segregated within the layers of CA3. And ACS seems to have different effects on different parts, but I guess that must be possible because of the kind of receptors you have, but I'm not aware of all the molecular biology details here, but anatomically there is a segregation. So high ACS suppresses recurrent connections more than inputs coming from EC, and high ACH also increases LTP, which we know from all the LTP studies on the CA3 recurrent connections. Okay, so the kind of thing, so and obviously under low ACH conditions, the opposite things take place. So I don't have to state that. So basically, ACH controls the two modes: high ACH you store and low ACH you retrieve. Right, and like I said, ACH is released by this cluster called nucleus basalis, which projects to the lots of cortical areas, and another structure called medial septum, right, projects to the hippocampus. Okay, so now ACH controls all the two modes, but what controls ACH? How does the hippocampus know, or ACH cluster, medial septum know when to release ACH or when not to release? Right? How does the brain know when to store and when not to store? Right, and here again, Hasselmo and all, they have an interesting theory. Because there is this medial septum and CA3 are connected in a loop like this. Medial septum is the cluster which releases acetylcholine to CA3, and CA3 projects to CA1, and interestingly, CA1 also receives direct input from EC, and CA1 is the one which projects back to medial septum. There's a negative feedback. So the Hasselmo and colleagues proposed that this loop is the one which decides when ACH has to be released. So the idea is that CA1 has access to the output of CA3 and the external input coming directly from internal cortex, EC. So it's in a position to compare these two. So the idea is CA3 is like a storage unit. It outputs the retrieved pattern from that storage unit from hippocampus. CA1 receives direct input from the external world. External world means from cortex to EC, right? That's what it receives. It compares these two. If the two match, then CA3 already has that information. It already has that information stored. So then it sends a signal to medial septum saying that I know this stuff already, so don't release ACH. Okay, there's no need to store this. Similarly, if on comparison, if it says that the two are different, then CA3 sends a signal to medial septum saying that, hey, this is new stuff. I've never seen this before. I would like to memorize this. So release SEH. So this difference that CA1 compute between EC output and CA3 output is used as a cue to trigger SH release from real setup. This is the idea, right? So there's a very interesting theory, but one thing that this theory misses is that just because a pattern is novel, I've never seen it, right? That doesn't mean I'll go ahead and store it, right? Because imagine you go to some marketplace, a busy marketplace where you see like thousands of new faces, right? Just because a face is new, you don't go ahead and remember. Very often you don't even remember your hostel mates, right, who you might be seeing once in a while, right? Because if you end up memorizing everything that you come across in the world, I mean, your memory system will be flooded very soon. So you need to have some notion of saliency. Like, is a given pattern, is it worth remembering, right? Only first you have to evaluate that, and then you have to decide whether to operate all this machinery to go ahead and store that pattern. Otherwise, you'll just let that pattern pass. You don't memorize it. You just let it flow through. So I don't see that aspect in Hasselmo's theories, at least as far as the way it is covered in this book called Gateway to Memory by Gluck and Myers. So I think we need a more comprehensive theory of, for example, memory operation. Like in my lab, we are developing an alternative theory where you don't need to think of CA3 as a memory unit. Because what is happening, what I've been noticing is, for a long time, people have been building this idea of CA3 as a memory unit. And if people have even calculated the memory capacity of CA3. So I still remember, one estimate which says that CA3 can store something like 36,000 patterns. Now, how the hell can you ever verify something like that? Right? Suppose there is a small circuit or a cluster of cells deep in the brain, and if you make a prediction that this cluster of cells can store some 10,000 patterns, how will you ever test something like that? So what's the point in making that kind of a prediction? So anyway, my gut feeling is that people have gone overboard. A lot of this line of thinking about hippocampus and CA3 has started with David Marr, who made some early speculations on that, and then people caught up on that and kept on developing theory. I feel it's possible to come with a more natural theory, more realistic theory than this. We've been trying to do something like that, and we still haven't published it. It's still kind of under construction. The work is going a bit slow. Okay, so that completes our discussion on hippocampus.